keep going. So the next class of molecules, macromolecules we're going to talk about are proteins. All right, proteins. Where do we get proteins from? Those are the building blocks. So where do we get them in terms of our diet from? Proteins. Mostly meat, right? What other sources are there for protein? We have beans, we have some leafy vegetables, right? Eggs, which are, I guess, animal source. Now, there's a lot of controversy about the source of proteins. Some persons, vegans or vegetarians, think that you don't need animal sources to get your proteins. Some persons think that you do need animal sources. So there's a lot of controversy as to what's the right or what's the best source of protein. Whether you get it from animal or non-animal sources, proteins are vital. They're very important for the body function. They form a vast array of structures and functions, and they are very, very important. Proteins are enzymes that catalyze metabolic processes. Proteins are hormones, such as insulin. Right? Proteins are transporters, such as hemoglobin. Proteins are receptors, and they're very important in cell signaling. All right, now proteins are polymers of amino acids. So amino acids are these small building blocks that link themselves together to make proteins. There are tons of different amino acids. Some are essential. Question? Some are essential, some are non-essential. What does essential versus non-essential mean?
Essential means we need to take it in from the outside.
Right, perfect. So essential means that we cannot make it in our body. We have to take it in from the outside, either through diet or some other type of supplementation. So we have to get those outside of our body. So some are essential, some are non-essential, and they play differently together to form all different types of proteins that differ in structure and function. Okay? All right. So the peptide bond is the covalent bond between the carboxyl group of an amino acid and the amino group of another amino acid. So I'll highlight what I mean by that here. All right, so an amino acid has an amino group on one end, right, which is this nitrogen and carbon, and it has a carboxyl group on the other end, which is this carbon double bound to an oxygen. Now, the carboxyl group of this, the carboxyl group of this amino acid and the amino group of this amino acid, right, the adjacent amino acid, will come together. They will release water. So this is combined with this hydrogen. They release water, and then they join together, and the peptide bond is what links these two amino acids together. What did we call that reaction earlier? What did we call that reaction? Condensation. Perfect. So these two monomers, or these two amino acids, are linked together. Water is released, and that we call condensation. So what we have now is a peptide bond sitting between these two amino acids. And this is how we make several different lengths of proteins. If we have fewer than 50 amino acids, we generally call this a peptide. And if we have more than 50 amino acids, if we have 100 or 150 amino acids, we call this a protein. Now, proteins have 4 different levels of structure. We're going to go through them individually. There's a primary structure, a secondary structure, a tertiary structure, a quaternary structure. And this is basically building up the complexity of the structure of these proteins. So the basic or primary structure or primary bond between two proteins is called the primary structure. And this is essentially what we just looked at, the peptide bond. So one amino acid forms a peptide bond with another amino acid, forms a peptide bond with another amino acid, and so on and so forth. So we have a long chain of amino acids. And that is the primary or the basic structure. Now we can get more complex and we can build upon this. The secondary structure is where we have hydrogen bonds being formed between the oxygen on this carboxyl end and the hydrogen on this amino end. So the hydrogen here and the oxygen here will form a hydrogen bond, And so what we get is several different twists and turns in this primary structure. All right, so just to remind you, the primary structure or the peptide bond was this peptide bond here in blue. Whereas if we complex it, or if we make the structure more complex, we can have this helical formation, which is what we're seeing here, and that is forming the secondary structure. Two examples of this are the alpha helix and the beta pleated sheets. Right, so the alpha helix on the left, which is just a helical configuration, and then the beta pleated sheet is a sort of hairpin or hairpin loop configuration, which is on the right.
And you said the hydrogen and oxygen were a hydrogen bond?
They were hydrogen bonds. Hydrogen bonds. All right. Any other questions? So this is the secondary structure. Let's look at the tertiary structure. So we start to get a little bit more complex here. We can have several different helical structures. So we can have helical structures here. We can have a beta pleated configuration here. And this is where the protein starts to get much more complex. And this looks like a jumbled mess, doesn't it? Does it look organized at all? It doesn't, right? So I'm seeing some head nodding. It doesn't. It looks quite messy. But in fact, it's actually quite organized. Okay, so there's lots of organization, lots of specificity in the tertiary structure of the protein. So much so that if there was an off structure, if anything was off even slightly, this would alter the entire function of this protein. This could lead to a loss of function. Let's say this was a receptor or a hormone. This could lead to a loss of function. This could also lead to a misfunction. So the protein or receptor could be doing something that it's not supposed to do. So this is very important, and in this jumbled mess is a lot of organization.
I was actually watching a documentary where they are trying to create proteins and predict what it's going to look like so they can, have artificial proteins.
Yeah, that's a thing now. So you can sort of
run a computer program to.
Right, yeah. So you can use or predict using the certain characteristics of certain amino acids, and you can put those together and sort of predict what this protein would do, what it would look like, and how it would function, which is really neat, actually. It's going to, I'm sure, develop later on. Lastly, any other questions before we continue? Lastly, we have the quaternary structure. The quaternary structure, and this is perhaps the most complex structure of this protein. Right, this is the where we have several subunits, lots of different tertiary structures underneath, and they're coming together to form one nice, big, organized protein. Okay, does anyone know what protein this is? Hemoglobin, right? So that's hemoglobin. You can see the 4 subunits. And hemoglobin is an important protein in the body. What is its function? Does anyone know? What is its function? Hemoglobin.
Blood.
So it's in the blood. What does it do?
Oxygen.
It carries oxygen. So it can carry around oxygen in the blood. Perfect. If we have even a slight loss of structure, or a slight misshape of this protein, we can lose the capacity to efficiently carry oxygen, right? That's very important. And this really underscores how that form follows function is important, very, very important on a small level and on a large level. All right, any questions on proteins? Any questions on proteins? Now we can have other different variations, more complex variations of proteins. We can have glycoproteins where you have a carb or carbohydrate added to that protein. That's important in cell recognition. It's going to be on the cell surface. We can have a lipoprotein where you have a lipid attached to the protein. And that's going to be important in transporting lipids. All right, let's move on to our last category of macromolecules here, the nucleic acids. And these are very important building blocks of our genetic material. Now, when I say genetic material, that's our DNA and our RNA, which combine to form what? What do these form? What does DNA and RNA form in the body?
Cell or our genome or
Like genes, right? So they form our genes and our genome. So they're very, very important. They are responsible for making you look uniquely like yourself, your eye color, your hair color, your height. All right, so these are very important building blocks here. Now, the structure of a nucleic acid or nucleotide is that they have a phosphate group. All right, so we got a phosphate group on the left here. It's connected to a 5-carbon ring, which can be either a ribose or a deoxyribose. Now, deoxyribose only has one less oxygen, right? Hence the name deoxy. And then there is a nitrogenous base attached as well. So we have a phosphate group, a 5-carbon ring, and a nitrogenous base. And we say nitrogenous base because these have nitrogen in their structure. Now, these bases can be either pyrimidines or purines. Pyrimidines or purines. The pyrimidines, which are on top here, have a single ring in their structure. Those are going to be cytosine, thymine, and uracil. So they have one ring, and these form the pyrimidines. Alternatively, the purines, which are adenine and guanine, have 2 rings in their structures. Okay. Now, as I mentioned earlier, the difference between DNA and RNA is the structure of the 5-carbon ring. So there's one less oxygen on the deoxyribose, whereas the ribose has an extra oxygen. Right? So we have deoxyribonucleic acid, which is DNA, versus ribose nucleic acid, which is RNA. And just to give you a bit of a recap, genes, which are coded for in DNA, eventually translate into proteins, and this is how we control different structures. So you may make different proteins because of your unique genome. You may make different receptors, different hormones, and so on and so forth. Now, nucleic acids also have a role in energy transfer, a very important role. This is something we're going to continue to see throughout this course. They're going to be important in capturing energy. So it's important to understand energy cannot be made. Right? This breaks what law? What law does this break? We say that somebody made or something made energy. Can we make energy? Can we make energy from scratch? Can I just make energy? No, what do I have to do? Right, so it has to be transferred from one to another. It has to be transferred from one form to another form, or harnessed in some way. Now, what is that law? What do we call that law? The law of thermodynamics. Thank you. So the law of thermodynamics says that we do not create energy, but we merely transfer it from one form to the other. So basically, adenosine can be bound to a phosphate group. That's where it's going to store some energy. It can be bound to 1-phosphate, adenosine monophosphate, or AMP. It can be bound to 2 phosphates, adenosine diphosphate, or ADP. It can be bound to 3 phosphate groups, adenosine triphosphate, or ATP. And we're going to see that molecule a lot throughout this course. It's going to be the source of energy for a lot of our metabolic reactions and different processes. Okay? And that adenosine is basically adenine plus the ribose, which we saw earlier. Now, also we have nicotinamide adenine dinucleotide, right? Nicotinamide adenine dinucleotide, which is NAD, as well as flavin adenine dinucleotide, which is FAD. And these are forms of nucleic acids that can bind hydrogens. And hydrogens are carrying electrons, which are energy. Okay, so the fact that they can bind hydrogens means that they can transfer or harness energy. They can create electric gradients, which we'll see when you look at some of the respiratory processes. And we do cellular respiration next week. They're going to be very important in harnessing energy in creating electric gradients that can generate ATP.